Assalamu alaikum and hello everyone. My name is Muhammad Davar Zahid and I'm a first year medical student at the Aga Khan University, and in today's video we'll be talking about the endoplasmic reticulum. In our previous two videos we talked about the nucleus and then we talked about the mitochondria, and I would suggest please do go and watch them so that you're caught up with everything that we're going to be talking about, and the concept you're able to build up a clear concept from the first base. Okay guys, so talking about the endoplasmic reticulum, it's a pretty simple structure. However, it sort of confuses us. So let's just take it slowly. So it's going to be an elaborate three-dimensional system of sheet-like membrane. And let's break this down. So basically elaborate. Why do we call it elaborate? The reason for that is that it's basically from the outer nuclear envelope all the way till the cell membrane. So it's basically spread out throughout the cell. And obviously we do have variations in certain cells. It's like really small. In certain cells we don't have endoplasmic reticulum, so it really depends. However, for the scope of our syllabus, we say that's a really elaborate structure, and I'll just show you guys a diagram as well, and in that diagram you can also see how huge it actually is. 3D. So it's 3D because essentially every single organelle and the entire cell. So we've seen the cell like this, right? A two-dimensional thing. However, the cell in reality is a 3D, it's an entire 3D universe. And I do have a class on this, in which I help you guys visualize how the cell looks. However, for now, essentially, let's combine these 2 points together. So sheet-like membranes. So what this means is, let's take pink. It's sort of like this, like these membranes. And they're three-dimensional, so they're extending like this, and it's a bit difficult to show it to you guys over here. However, I do have like a diagram and I'll show that to you guys as well, and it'll all be clear. So we are done with like a brief introduction of the endoplasmic reticulum. Let's move on. Okay, so talking about the different types of endoplasmic reticulum, we have the rough endoplasmic reticulum and we have the smooth endoplasmic reticulum. ER stands for endoplasmic reticulum. So rough means it has ribosomes present, which gives it a rough appearance on a microscope and in a microscope, and this ribosomes absent. It's pretty simple. That's why it's smooth. Okay, talking about the functions, so let's just talk briefly about their functions, and then we can go and we can see a structure of these endoplasmic reticulums. So the first thing that we have is that the ribosomes, so we'll be talking about this in our protein synthesis chapter. The ribosomes are involved in what is known as the protein synthesis. So because rough endoplasmic reticulum has the ribosomes, we have protein synthesis going on. And one more thing that we have is mechanical support. So essentially the structure is so big, and the structure, these membranes, they're spread throughout the cell. So it's able to give the cell sort of a mechanical support. In the same way, if I take, let's say I take a basketball, and in one basketball, I basically put sort of like this metal sheet, and in one basketball I put nothing, and I deflate both of these. So previously it was like this because the metal sheath one had this metal sheath. It's still sort of going to be able to retain the the shape because there's like a metal sheath inside of it, sort of like this. However, in the ball that has nothing, it's just air, and once that air is out, it's going to be deflated like this. So that's what the metal sheath does. And the metal sheath in this case, talking about the rough endoplasmic reticulum, it's sort of like, this mechanical support, because the structure itself is so huge. It's, from the outer nuclear envelope all the way to the cell membrane, it sort of, gives us this mechanical support. And now let's move on to the smooth endoplasmic reticulum. So the smooth endoplasmic reticulum, what it does is that the first thing is, so it actually has more functions. It's involved in the metabolism of carbohydrates. It's involved in the detoxification of drugs. And also poison. And it's particularly important in the liver cells. So if you guys plan on, pursuing medicine or a career in healthcare which has a pharmacology course, you'll be discussing, the first pass metabolism and all of these things. And over there you'll learn that the liver cells, the hepatocytes, they're involved in detoxifying or, in activating drugs that we take. So that's pretty important because they love to give you questions on this. And then we also have synthesis of lipids. And there's a particular class of lipids that we have in our body, and these are called steroids. And they're sort of involved in all of our, different, what do we say, sort of like cell signaling, hormones, all of these things. And we'll talk about this when we're talking about the biological molecules chapter, but that's what we have. And one more thing, that's calcium ion storage. And this calcium ion, it's involved in, nerve impulses, all of those things, not that important right now. And the fifth thing is transport. And I will have to pause this for now. This transport thing, we won't be talking about this right now, because I want to teach you guys the cytoskeleton, the cycloses, all of these things. Once you've done ribosomes, then we'll talk about transport. And lastly, it also has mechanical support. So both of these structures, because they're so big, they're going to have mechanical support as, a basic requirement. So that's what we have for the functions and the types of the endoplasmic reticulum. Okay, so let's try and visualize the endoplasmic reticulum. I have a diagram on board, but before that, before I start to explain it, I want you guys to pause the video and I want you guys to try and make sense what's happening in this diagram. Okay, so I hope you guys have done that. So in the start, we have this, this nucleus present, and I told you guys that we have the endoplasmic reticulum extending from the outer nuclear membrane. So that's the case. It's basically, if I look in a microscope, it sort of looks like they're joined together. They're that close. However, as we know, they're two different structures, but we have the nucleus, and the nucleus has these nuclear pores present. And I talked about these nuclear pores in our video on the nucleus, but we can clearly see them over here. We have the nuclear pores, we have the nuclear envelopes. Now talking about the next thing. So we can see that we have these sheets of membranes. A, B, what we can see is that we can see these ribosomes, right? So if they are sheets of membrane and if we have these ribosomes, that means that we're dealing with the rough endoplasmic reticulum. So let's just quickly shade that. I'm sorry this is so crude, but I'm not here to color between the lines. Okay, so we have the rough endoplasmic reticulum present. Let's just take that. And then the next thing that we see are these weird tube, it's sort of like tubes. Don't worry that they look like tubes, they don't look like tubes. So and at our level, all that they want us to see is that the rough endoplasmic reticulum has ribosomes. These things, these tubes, whatever they are, they don't have ribosomes. That's all. And they're pretty easy to identify. I'll be doing a few questions with you guys as well, and we'll be able to identify how the smooth endoplasmic reticulum looks like. So let's take that as well. Peripheral sheet, whatever this is, we're not concerned with that. So ribosomes, we can see them on the rough endoplasmic reticulum. Okay, let's talk about the cisternal space and the cisternae. So essentially, these membranes, they go like this, and they're just going on and on and on. In the middle of them, we have these small cavities of sorts, so small spaces, which are called cisternae. So these enfoldings, if you may, these enfoldings, they're called as cisternae. That's all. These cisternal spaces, don't really concern yourself with that. We only need to know about cisternae. Cisternal spaces are basically the space inside the cisternae, that's all. So we have these enfoldings, and let me also write that down over here for you guys, because structurally that's really important. Cisternae, these are enfoldings of endoplasmic reticulum membrane. Okay, and these are present in both, just by the way. Okay, and now, just a second. Okay, so now we are done with the endoplasmic reticulum. Okay guys, so just like I said, we just covered the entire endoplasmic reticulum. There are just two, three small things which are pretty high yield for your A-level exams and also for all of your future exams. That is that the number of endoplasmic reticulum will vary. And I mentioned this briefly before, but let's just do it in a bit of detail. And really simple. So basically, if I have a cell that is secretory, I have a lot of proteins being made, or let's say I have a lot of steroids being made, a lot of whatever, so it's like secretory in nature. Or let's say that I have a cell that is involved in the detoxification of drugs, which is a liver cell. So both of these cells are going to have a greater number of endoplasmic reticulum compared to a cell which, let's say, does not have a lot of protein synthesis, does not need a lot of, it's not like a glandular cell, it's just a random cell, so it won't need it. So it won't have it'll probably have like a really small endoplasmic reticulum. It won't have these elaborate, like, do you guys see? I want you guys to appreciate how huge this actually is. So like if I make a normal cell has a nucleus over here, so that's pretty much what the size of the nucleus is. So can you guys compare these both? So like if I draw it out really roughly, let me just use like a pointer, so it's going to be all over here. So it's really huge, but in certain cells we don't need this, so it's small. However, in these two types of cells we have a lot of endoplasmic reticulum present.